Okay, our last objective here is to identify the myelin sheath and describe how it is formed in the CNS and the PNS. So let's talk about myelin. So myelin is basically the layer of fat that insulates our neurons, right? Insulates our nervous tissue. So in the central nervous system, myelin is produced by oligodendrocytes. So here's what an oligodendrocyte looks like, or sorry, here's what an oligodendrocyte looks like. It would be in the, I'm sorry, that was correct. So here's what an oligodendrocyte looks like. Here is the myelin that's being laid down, and the oligodendrocyte is going to send these extensions to several axons at a time. So notice this one cell can actually myelinate more than one axon at a time. It's going to produce myelin along these neurons, but it's not going to produce any neurolemma. I'm going to talk about what neurolemma is, but neurolemma is only seen in the peripheral nervous system as a structure formed through myelinating peripheral nerves. And we'll talk about what that is coming up. Myelin is what gives tissues their white color. This is what we refer to white matter as being white. And that is essentially fat that's being laid down as myelin. And then gray matter is where we have the cell bodies or the somas of these neurons. And so that is not going to have any myelin. And so we're going to refer to that as being gray or gray matter. All right, so gray matter is unmyelinated, the somas and cell bodies of neurons. White matter is the myelinated axons of those neurons. And another important thing to point out here, and you'll probably talk about this in the lab, is that the white matter and gray matter are going to be reversed from the brain to the spinal cord. So if we look at the brain, gray matter is going to be overlaying the white matter. That is, the somas and cell bodies of the neurons that form the brain and the cerebral hemispheres are going to be on the outer surface, whereas the myelinated portions are going to be internal. If we look at a cross-section of the spinal cord, we're going to see that being reversed. So the center of the spinal cord is going to have gray matter, and the outer portion of the spinal cord will have white matter. Okay, those myelinated axons. Let's look at what neurolemma is. So in order to talk about neurolemma, we need to consider the Schwann cell, which is the main cell that myelinates in the peripheral nervous system. So think about your peripheral nerves, everything outside of brain and spinal cord. That myelin is going to be laid down by the Schwann cell. And it's going to begin with a structure called the neurolemma. So basically, we have a Schwann cell. So here's the Schwann cell. Here is the nucleus of that Schwann cell, this large brown structure. And then here is the neurolemma. Now, the neurolemma is basically the projection of the Schwann cell that then begins to lay down the myelin around that axon. So the structure in blue here, here's a cross section of that axon that we're trying to myelinate. And then the projection of the Schwann cell that begins to lay down that myelin sheath is what we refer to as the neurolemma. Here is a slightly different stage in the process. So here we can begin to see myelin already covering the axon. Here's a different view of that. We can actually see the axon in the middle and the myelin covering that. And then notice the Schwann cell is going to still be intimately connected to this neuron, but then the myelin will be in between the axon and the Schwann cell itself. That is to say that the Schwann cell is not, the Schwann cell creates the myelin, but the Schwann cell is not the myelin, right? I want to make a distinction between what is the Schwann cell and the myelin that's actually being laid down in between the Schwann cell and the axon. Okay, and then another point to make here is that, again, we can myelinate several axons at a time. It doesn't have to be one by one, just as we saw with the oligodendrocytes. And here's what it looks like when we have a single Schwann cell, again, this really large nucleus being that of the Schwann cell, and then several axons in blue here, and then several myelin sheaths being laid down by the neurolemma of this Schwann cell. Okay. Now, I do also want to make the distinction that myelin is laid down, but then we can also see neurolemma in unmyelinated axons as well. So for example, even though we have, so here's a myelinated axon. Here is that actual myelin sheath that has been laid down. This is the darker gray structure. And then here's the cytoplasm of the Schwann cell that's still sort of intimately wrapped around this axon. However, notice in the periphery, oops. Okay, notice in the periphery, we have another unmyelinated axon here. And then it's still going to have the projection around the layer of this axon will still be considered neurolemma. So neurolemma is not synonymous with myelin. I want to make that clear, right? So it's the Schwann cell, the neurolemma being the projection of the Schwann cell, and the myelin being the fat that's being laid down around the axon itself. All right. Okay, let's pause here and see if there are any questions first.